Hi everyone, today we are going to understand the topic DNA replication. DNA replication is a process by which DNA makes a copy of itself during cell division. It is a semi-conservative, bidirectional and discontinuous process. Semi-conservative method of DNA replication was found by Messelson and Stahl in 1958. DNA replication is an essential mechanism in enhancing cell growth, repair and reproduction of an organism. Before understanding the mechanism, we will see what are the enzymes involved in DNA replication. First one is DNA helicase. DNA helicase is an enzyme which helps in unwinding of DNA and separate the two strands of DNA by breaking hydrogen bonds between them. DNA primase. It is an enzyme which helps in the synthesis of short RNA primer, providing the starting point for DNA polymerase to begin building the new strand. DNA polymerase. It is an enzyme which helps in the synthesis of new DNA strand by adding complementary nucleotides in the direction of 5' to 3' direction. DNA ligase. It acts as a glue that helps join the synthesized DNA fragments together on the lagging strand. Topoisomerase is the enzyme which relieves the tension and prevents supercoiling that builds up ahead of the replication fork as the DNA unwinds. Single strand binding proteins. These are the proteins that coat the separated single strands of DNA to keep them away from each other and avoid again coiling of the DNA strands. Now we will see the mechanism of DNA replication. The mechanism of DNA replication involves mainly three steps, that is initiation, elongation and termination. Initiation is the first process in which the two strands of DNA unwind at the region of origin of replication, and the enzyme called as helicase opens the DNA at the replication fork is formed at the point of separation of strands. DNA is coated by single strand binding proteins around the replication fork to prevent rewinding of DNA. Topoisomerase prevents the supercoiling of DNA and release the tension. RNA primers are synthesized by primase complementary to the DNA strand. Elongation. DNA polymerase starts adding nucleotides at the end of primers. The leading and lagging strands are formed along the parental strands, and the lagging strand contains the fragments of DNA called as Okazaki fragments. The leading strand is formed in the direction of formation of replication fork, and the lagging strand is formed opposite to the direction of replication fork. So in this diagram we can see that this is the parental strand and this is another parental strand. Here we can see the helicase is unwinding the DNA strand and continuous DNA is formed, which is leading strand, and here the discontinuous DNA fragments are formed, which is lagging strand. As we can see, the leading strand is forming in the direction of formation of replication fork, and the lagging strand is formed in the opposite direction to the replication fork. There are different components like topoisomerase, single strand binding proteins, DNA primase, which is involved in the formation of RNA primers. These are the RNA primers, and we can see DNA polymerase, and this is the lagging strand, this is the leading strand. Next we'll see the termination process. Termination occurs when the two replication fork meet at the termination region of the DNA molecule. DNA polymerase completes the synthesis of the remaining DNA strands, and the RNA primers are removed and replaced with the DNA nucleotides, and the DNA ligase seals the gaps. Topoisomerase separates the newly formed daughter DNA molecules, resulting in two identical DNA molecules. And at the end of replication, we will get two strands of DNA, which contains each strand contains the one parental strand and the another newly formed DNA strand. Thank you.